A pleasant day to you, icons and lady icons. Welcome to the fourth module of our discussion in the subject General Biology 1, and in this topic, we are going to discuss the cell's intricate machinery. Have you ever wondered what are actually the different things that is happening within our bodies, especially on our cells? And in this module, we are going to have a short overview of the different things that is happening within every single one of them, within every single organelle that is present on us, humans. Specifically, we are considered as eukaryotic organisms. We are under the domain Eukarya, specifically the kingdom Animalia, and we're also under the phylum Chordata. And since we are considered as animals, there are actually different mechanisms that is happening within every single parts of our cells. But once again, this will just be an overview of all the discussions that we are going to have for modules 5 onwards, since we are going to talk about still the different mechanisms that is involved in cytology. On the previous modules, we discussed here the concept of life, and at the same time the building blocks of it and even the basic unit of life. Now, we're actually going to apply those concepts in this particular module, and let's have a short review. So once again, we humans are considered to be as eukaryotic cells because we are possessing nucleus, the endomembrane system, energy processing organelles, and at the same time organelles for structural integrity, movement, and communication. But I'm actually going to provide here a website for every single one of you to access other than the discussions that are present on your modules, for you to see what are actually the different characteristics that is present on the different types of eukaryotic cells, specifically animal cells and plant cells. On our discussion for today, we are just actually going to focus on two major parts, specifically the endomembrane system and the organelles for structural integrity, movement, and communication. The endomembrane system, this is actually one of those organelles that makes eukaryotic cells different from prokaryotic cells. Obviously, other than the presence of the nucleus, which is the most conspicuous part of the cell, we're actually going to consider here that in the case of the endomembrane system, the nucleus is still responsible for the different things and mechanisms that will simply happen in this part of the cells. And just a short discussion regarding the nucleus, always remember that the nucleus is responsible for containing our genetic information in the form of the DNA. And there are actually different types of materials that are still present on your nuclear envelope, specifically on the nuclear membrane. Then, in the case of the energy processing organelles, namely the mitochondrion and the different types of plastids, specifically the chloroplast, we're going to have more discussions regarding this matter when we discuss bioenergetics on the second quarter of General Biology 1. But once again, what would be the highlight of our discussion for today? It would be the endomembrane system and the organelles for structural integrity, movement, and communication. In this endomembrane system, that is simply created because of the process known as infolding. Because of the presence of the different folds of the plasma membrane that is present on the ancient eukaryotic cells, it actually created here a series of membranes that are responsible for the formation and synthesis and even breakdown of various biomolecules. And in the case of the endomembrane system, it is actually consisting of different parts. Now, we can observe here the presence of the nucleus that is present at the center. Please take note that the nucleus is not a part of the endomembrane system, but rather the endomembrane system will start with the presence of the endoplasmic reticulum. Endoplasmic reticulum, it is the part of the cell, specifically of the endomembrane system, that is the most adjacent to the nucleus, or this is very near to the nucleus. And the endoplasmic reticulum is made out of two major parts. The rough endoplasmic reticulum, which is containing now the ribosomes, and the smooth endoplasmic reticulum that is present on the farther region of the endoplasmic reticulum. Rough endoplasmic reticulum is usually nearer to the cell's nucleus, since the rough endoplasmic reticulum, once again, it is containing ribosomes. And remember, when it comes to the nucleus, it is containing the genetic information known as the DNA. And because of the presence of the RNA, mRNA, tRNA, and rRNA, the central dogma of molecular genetics is happening, and its last stop is the ribosomes, and some of those ribosomes are attached on the rough endoplasmic reticulum. While in the case of the smooth endoplasmic reticulum, it is now present on the farther region because it is now responsible for the distribution and the movement of the different biomolecules. Smooth endoplasmic reticulum, it is also responsible for the formation of the other parts of the endomembrane system, namely the presence of the different vesicles. These vesicles, which contain different materials that is simply created by the endoplasmic reticulum, it will now be transported to the Golgi apparatus. This is actually a misconception whenever we are talking about the presence of the endomembrane system. Please take note that the endoplasmic reticulum, both rough and smooth endoplasmic reticulum, they are not only passageways, but rather they're responsible for the formation of the different materials and even biomolecules that are present within every single one of us, within our cells. In this endoplasmic reticulum, different types of biomolecules, namely proteins, carbohydrates, and even lipids are synthesized and broken down in these different regions of our cells. And if we're going to consider this, the rough endoplasmic reticulum, it is responsible for anything that is related to proteins, while the smooth endoplasmic reticulum is responsible now for the formation of lipids and even some carbohydrates that will be utilized by our body. Remember, endoplasmic reticulum are not highways only. They are not passageways only, but rather they are responsible for synthesis and breakdown of these different types of biomolecules. Then, from the smooth endoplasmic reticulum, vesicles will now be created, and this will now be transported on the Golgi apparatus. Now, whenever we are talking about the different types of vesicles, we actually have here two major types of vesicles. We have the presence of transport vesicle, and we also have here the presence of the secretory vesicle. Now, let's focus first on these vesicles. Transport vesicles are responsible for transporting different materials within the cell. So, for instance, this transport vesicle is now transporting different types of biomolecules that were created on the endoplasmic reticulum towards the Golgi apparatus. While the secretory vesicles, they are now responsible for moving in and out of the different materials that were simply created by either the endoplasmic reticulum or by the Golgi apparatus. There are actually other types of vesicles, namely the vacuoles. Vacuoles, they are now responsible for storage, okay? Vacuoles are for storage. Vacuoles may contain food materials. They could also contain waste products and also the presence of water. Then, there are also other types of vesicles. Specialized vesicles containing enzymes, which is responsible for the digestion of different food products, namely known as the lysosome, and even the presence of peroxisomes. So these are actually the different specialized types of vesicles, but we're actually going to discuss them on the later part of the discussion regarding the endomembrane system and its mechanisms. Now, let's focus on the Golgi apparatus here at the center. The Golgi apparatus, it is not only responsible for packaging, just like what is simply discussed to you probably during your Science 7 or even on your Science 8. But remember, the packaging center of the cells are not only responsible for packaging, but rather they are responsible for modifying the different biomolecules that were created by the endoplasmic reticulum. As you can observe here, different materials will simply be passed from the endoplasmic reticulum by the vesicle, then it will now enter the Golgi apparatus. The cis phase, it is now the entrance of the Golgi apparatus, and the different biomolecules will now be modified on the cisternae, and it will now be released on the trans phase. Okay? Remember, cis phase, it is the entrance. Then the modification center, it is the cisternae, and the exit, it is known as the trans phase. Okay? And these materials will now be removed by the secretory vesicles, or they could be stored on different types of vesicles known as vacuoles, or they could also contain enzymes and other materials, even the presence of hydrogen peroxide, just like on peroxisomes, then enzymes that is now present among lysosomes. Okay, so that's the thing whenever we are talking about the different parts of the endomembrane system. Now, how are we going to apply these concepts? For instance, we have here the presence of the transformation of the different materials, just like the transformation of proteins. We have here the presence of the amino acids. So please follow the cursor that you can see here on the screen. We're going to start with the presence of amino acids. Okay, and remember, amino acids, these are the monomers of proteins. Then, these amino acids will be manufactured and will be joined together by the ribosomes during the translation process of the central dogma of molecular genetics. Then, after the formation of the different chains of amino acids, it will now create polypeptide chains. Then, these amino acids and polypeptide chains will now be transported from the rough endoplasmic reticulum to the smooth endoplasmic reticulum. Then it will be transported by the transport vesicle towards the Golgi apparatus. And it will enter the cis phase. It will be modified on the cisternae, then it will be released on the trans phase. These amino acids, when they join together, they will now create polypeptide chains. And these polypeptide chains will be modified by the Golgi apparatus to create now a specific type of enzyme known as amylase. Okay, so if you're aware what is actually an amylase, it is the enzyme that is present on our saliva, specifically salivary amylase. Okay, the enzyme that is present on our saliva, it is known as amylase. It is responsible for breaking down polysaccharides to disaccharides. The reason why we can taste sweet, sour, bitter, salty, and even the presence of umami taste, it is because of the amylase, and that is present on our mouth. Since this amylase will simply be modified by the Golgi apparatus, it can now be released by the secretory vesicle, and we can now see that our body is actually secreting saliva. Okay, as you can observe now, at this very moment, probably some of you are salivating, probably some of you are hungry, or some of you are just daydreaming at this very moment during my discussion, and saliva is being released on your body. So definitely, it is just created because of the different presence of the amino acids. Then, the polypeptide chains after the formation of these polymers will now be modified on the Golgi apparatus and will be released by the secretory vesicles. So, other than proteins, I also have here another example. For instance, we have here the presence of lipids, specifically fatty acids. Okay, so fatty acid monomers will now be responsible for the creation of steroids. Then steroids, there are actually different types of steroids. Okay, remember, steroids, it is just a general term that we are using for different things that is related to different types of fats. Okay, these steroids will now move from the smooth endoplasmic reticulum to the Golgi apparatus by means of the transport vesicles. Then, it will now be modified on the cisternae, then it will now release androgens. What are androgens? Androgens, these are sex hormones, specifically testosterone or estrogen. Okay, these androgens are simply different types of steroids that were made out of the hydrocarbons that are still present on fatty acids. So you can actually see here the different modifications that will simply happen whenever we are pertaining to the concept of the endomembrane system. The endomembrane system is responsible for creation, then also modification and the delivery of these different materials. If the cells need androgen, it will not be released. But if the androgen is needed on the exterior part of the body, it will be released. And lastly, last example, this also involves proteins, okay? For instance, the enzymes that I am simply talking about earlier created now the digestive enzymes. Okay? If this digestive enzymes is needed by the cell, they will now be stored on lysosomes. But if it is needed to be released, for instance, on our stomach to break down different types of food products, specifically break down different types of proteins in our stomach, it will now be secreted on our cells. Okay? Once again, it is case-to-case basis. If the cell needs it, that material will now stay on the cell. If the cell doesn't need it and other parts of the body need that material, it will now be secreted or it will now be released. So that's the thing about the mechanisms of the endomembrane system. Once again, it involves the endoplasmic reticulum, then the different types of vesicles, then the Golgi apparatus. Then obviously, if it will be released with the plasma membrane. Now, there are actually specialized parts of the endomembrane system. Other than the presence of the lysosome, there's also the presence of peroxisomes. Peroxisomes, these are now specialized types of vesicles that are containing oxidative enzymes that is responsible now for helping in the breakdown of other materials. If, for instance, lysosome cannot break down the certain type of material, the peroxisome will be present for it to be responsible for aiding the enzymes that are now present on the lysosome. So there are actually different processes that is happening within our cells. For instance, we have the presence of phagocytosis or cell eating. But on the future modules, we're actually going to discuss here other types of cell eating and even cell drinking mechanisms other than the phagocytosis. But in general, for instance, we have your phagocytosis, in which if the cell will ingest food products, it will now be incorporated on a food vacuole. Once again, vacuole is responsible for containing different materials. Then, lysosome will merge with the food vacuole, and the digestive enzyme that is present in this lysosome will now break down the material that is inside this food vacuole. Then digestion will happen. In some cases, if the different parts of the cell were simply worn out or they are useless, they will be killed by the lysosome with the aid of the peroxisome. With the process known as autophagy or self-eating, it will now eat the useless part of the cell. For instance, in this case, we have the presence of a mitochondrion. For instance, this mitochondrion is no longer useful for the cell. It will now be digested by the cell, and it will now create the different monomers or even polymers that will now be utilized by the cells themselves, and it will now be recycled. From the name itself, autophagy, it is self-eating. You are now eating a part of yourself. And in this case, it is the mitochondrion.